Welcome to another edition on Mariner Biology. This one's going to be about the cell cycle, and we're going to spend some time on meiosis, which is a little bit more complicated than mitosis. But if you understand meiosis, no reason you shouldn't understand mitosis too. Okay, so again, we're talking about the cell cycle. And the cell cycle is composed of two big things, and then one of those big things is broken up into three things. Okay, so this first part that I've shaded of this circle that I've broken up into a bunch of pieces, that's what we're going to use to represent something called interphase. And the interphase is where the cell spends most of its time. It doesn't look like it's doing much if you look at it under a microscope, if you see a cell in interphase, but there's actually a lot going on. Okay? And the purple shaded part is called the M phase. Okay, so we've got three parts to interphase. There's one phase called G1, the second one's called S, and the third one's called G2. The last one is M, and that's where we're going to have either mitosis or meiosis, followed by the actual division of the cytoplasm and the cell, which is called cytokinesis. Okay? So let's look at the difference between mitosis and meiosis real quick. And like I said, after this, we're going to spend some more time on meiosis. So here's what mitosis and meiosis do for us. Right? Mitosis is really kind of this workhorse. It allows for you to grow, develop. You can repair or replace cells. If you get a cut, it's mitosis that's going to fill in that cut with new cells. If you are an asexually reproducing organism, that's what you got to use. Unless you're bacteria, they do it a little bit differently. There's something called binary fission. And then we also did a lab where we talked about the importance of having a lot of surface area for a little bit of volume. And if we divide the cell before it gets too big, we can maximize how much surface area we have for the volume. All right, and then we've got the main purpose for meiosis is sexual reproduction, right? Where we're going to make these things called gametes. And mitosis results in two cells after we have one division. Meiosis, we're going to get two separate divisions where we end up with 4 cells. So guys, you're going to end up with 4 sperm for every cell that enters into meiosis. Girls, you're just going to have the 1 egg. Okay? And you can tell here that we're in high school, so that egg wants nothing to do with that sperm. Okay? That's why it looks a little angry there. So let's look at the phases of the parts of interphase more closely. So we can see that there's a lot of stuff going on in the G1. The S is really important because that's where we actually duplicate the chromosomes so that we can have 2 sets for mitosis and we can have 2 sets to play with for meiosis. Okay, so you can go ahead and read all those after they come up. But the point is that there's a lot of stuff going on during interphase. It's not just the cell sitting there waiting to divide. All right, and then in the M phase, which is where we'll have mitosis or meiosis, at the end of either mitosis or meiosis, we're going to have something called cytokinesis. Okay, and again, mitosis and meiosis is where we're actually dividing the nucleus. Right, we're separating the sister chromatids back into separate chromosomes, and in mitosis, we want to end up with the number we started with. So if we start with a diploid number, if it's a human cell, we start with 46 chromosomes, we definitely want to end up with 46 chromosomes. Remember, we're really just trying to replace cells or add new cells, and we want those all to be identical. Okay, if you're in the testes or ovaries, you're going to do something called meiosis, right? So you're going to start with a diploid cell, but you're going to end up with 4 cells that have half the number of chromosomes. But again, we have to divide the nucleus up, and meiosis is going to do a little bit differently, we're going to see next. And then we got cytokinesis, which is again the division of the cytoplasm. And make sure you know the difference between cytokinesis in plant and animal cells. Alright, so this next slide, this is kind of what we did when we were trying to use those pop beads to model what's happening in meiosis. Okay, so if we go through the first part, let that all animate itself out there. Okay, so in the beginning, in the G1, we're looking at a cell that has a diploid number of 4. We have 2 long ones, a purple one and a light blue one, and then we've got a pair of short ones. Okay? Remember, these are called homologous chromosomes because we have 2 colors that represent 2 parents. So as long as we're inside a cell and that cell has 2 of each chromosome, 1 from each parent, then we're fine. And remember that the only reason those 2 chromosomes are not identical is because of there's the possibility that they could have different versions of the genes. They have the same numbers of genes, same kind of genes, they're the same length. Everything about them is the same, except for one may have a version of a gene that gives you a widow's peak, and the other one may have a version of a gene that gives you no widow's peak. Okay? So we go into the S phase, and we duplicate the chromosome. So we can see where we start with a physical number of 4, we now have a physical number of 8. But we have 2 identical copies for each chromosome. Those are called sister chromatids, and they represent 2 identical copies of the same chromosome. So we actually consider the 2 sister chromatids as 1 chromosome. Okay, so we still have 4 chromosomes, we just have 8 sister chromatids. All right, and then in prophase 1 of meiosis 1, the sister chromatids come together in pairs, and they're held together at the centromere, and actually all along their length, and then they form tetrads. And the tetrads are the sister chromatids of each homologous chromosome coming together, forming that foursome you see. And again, we're still diploid because we still have both parents represented. We still have homologous pairs of chromosomes. It's just each homologous chromosome is made up of 2 sister chromatids. All right, then something really special happens in the later part of prophase 1. It's called crossing over, where we actually switch a segment from each of the inside sister chromatids of the homologous chromosomes. So we get something called genetic recombination. So if one of those sister chromatids, when it separates and becomes a chromosome, is the one that gets into the sperm or the egg, and you end up inheriting that one, then you've inherited a chromosome that has a unique arrangement of genes on it that's different from either of your parents. And that's why you look different than your parents. And that's a really good thing. Okay, and then we get into metaphase 1 of meiosis 1, where the cells line up in the middle. And this is really important because we get something called a random assortment of chromosomes. We don't know if the blue sister chromatids are going to be on the right side of the cell or the left side of the cell, and same for the smaller ones underneath. So they assort randomly. So this and the crossing over we just talked about are really important sources of genetic variation. All right, so let's look at our last slide. So we have anaphase, right? The tetrads separate, the homologous chromosomes separate, but the sister chromatids stay together. And then we get into telophase and cytokinesis of meiosis 1. All right, we get 2 new cells. And nothing much happens between telophase 1 and cytokinesis and then going into prophase 2 of meiosis. All right, so I've drawn them so that the chromosomes look the same way. Okay? But now we're no longer, at the end of telophase 1, we're no longer diploid again because we don't have a long purple one and a long blue one. We've only got a long blue one and a short purple. Right? So now we're diploid. We don't have both parents represented for any given chromosome. Yeah, there's a long blue and a short purple, but it has to be both parents represented for the same chromosome. All right, so we go through prophase 2, then we go through into metaphase 2 where the chromosomes, the chromosomes line up, but this time what's going to happen is sister chromatids are going to separate. Okay, so watch this. So we have sister chromatids separating, right? And now these cells are going to divide after telophase and cytokinesis. And remember, we're totally haploid at this point, and we continue to be haploid, and what we're going to end up with is 4 gametes, right? 4 haploid cells, because remember, now you can see it's pretty clear now, each have 2 chromosomes, and we started back in the beginning of all this with 4 chromosomes. So we've cut the chromosome number in half. Okay, and we have 4 of these cells. And in girls, 3 of them are going to go bye-bye, and you're only going to use 1 as an egg. and in guys, all 4 of those are going to turn into sperm. Okay? And that's important because the reason we need to cut it in half, the number in half, is because when a sperm and an egg come together, right, 2 halves are going to make a whole again. So we're going to get back to that original chromosome number of 4, which is a good thing because we need all that information so that you can develop from a single cell into the healthy young person you are today. So I hope this helps, and study hard tonight. You got a test tomorrow, and come with questions because we'll have time to answer them before the test starts. Have a good night.